In my laboratory, we use two different types of approaches to study the PCDH19 protein. We tend to use hippocampal models of neurons that are being cultured and animal models. So we have two different types of models, animal models, we are working on. Right now, we have no PS cells that have been obtained from patients, but we would very much like to have them in future times. Now, as far as the molecular part, our primary interest is the one of understanding what's the function of protocadherin 19 in the neurons. And the most simple way to study it is to use cultured neurons. So the first thing we did was to go and look what's the temporal expression of the protein in neurons and at the same time in the animal. As you can see, in a very simple way, in this box, we show some homogenized and lysed hippocampal brain cortex areas, that is to say, two areas that are very important for the learning and memory processes, and in rats at the same time, but in a different stage of their own development. At the same time, in parallel, we also analyzed during synaptogenesis and in mature neurons the expression of this protein. So what is the course of this protein? What is the trend undertaken by the protein? You can see it from the Western blots, but even more so from the diagram. You can see that the protein is particularly expressed in a stage which is the pre-adolescent stage in the rat. And at neuron level, it expresses itself at the time of the maturation, that is before synaptogenesis. But nonetheless, the protein is well expressed also in the adult rat, but also in mature neurons. This is a very representative box showing that the protein in the adult rat brain is expressed both at the level of the cortex and at the level of the hippocampus, but not much at cerebellum level. The second question we wanted to answer was the one of analyzing at neuron level whether the protein expressed at excitatory synapses or inhibitory synapses level, whether it did express itself in one or the other. So let's say that we define excitatory synapses, those synapses that use as a release or a neurotransmitter glutamate, whilst the inhibitory ones are the ones where there is a release of GABA. So we then went to see in the cultured neurons the distribution of protocadherin at the level of the synapses. In order to compare it with the excitatory and inhibitory synapses, we used two specific markers typical of excitatory and inhibitory synapses. For the excitatory ones, we used the PSD95, and for the inhibitory ones, the alpha-1A subunit of the GABA receptor. Now, you can probably see, just looking at the images, that the protocadherin, this one, and here we did use an antibody which is specific for protocadherin. Well, as I said, protocadherin has been localized in given patches, which well localizes with PSD95, but also co-localizes in part with alpha-1. And the conclusion is that protocadherin is expressed in both types of synapses. So we know now where the protein is distributed. We know that it's present at the level of the hippocampus and the cortex, and we also know that it has a temporal expression. So we started playing a little bit. We wanted to modulate the protocadherin expression in a set of experiments, still in cultured neurons. We silenced, that is, we abated the expression of the protein. In a different set, instead, we were using two different increasing doses, or rather, we increased the expression of the protein, overexpressing it more and more. So what we saw, and here we have the Western blots, we prepare samples in such a way as to have some extracted homogeneated results that were extracted from the motor neurons after having treated them either by silencing or by overexpressing the protein. And then after that, we tried to see what happened at synaptic level, protein level. And what we saw is that in the absence of the protocadherin 19, there is a trend to see a reduction of the GABA expression. But the most interesting thing is that by just overexpressing the protein, we have an increase of synaptic markers, the PSD95, and of the alpha-1 subunit, and of the GABA receptor as well, meaning that the protocadherin is sort of modulating the expression of some synaptic markers that are involved in the neuronal development. What we then did was to go and see what was the undergoing mechanisms, the molecular mechanism underlying all this. And the way to understand it was to try and find the so-called interactors, meaning to try and see with which ones the protocadherin is interacting. So we saw that we tried to identify the new molecular patterns, and this is the structure of the protein. And by just using this cytoplasmatic tail, that is the cytoplasmatic part, we saw that it's interacting with the GABA, with the alpha-1 subunit of the GABA receptor, meaning that this assay was validated in different other ways. The simplest one is to go and see whether the two proteins in a non-neuron system, but rather in a heterologous cellular system, when brought together, could form a complex and whether they could associate themselves. And by using these experiments of immune precipitation, and this is how we carried out the assays in order to see whether the proteins did associate themselves. And we saw that while expressing the cells at heterologous level, they can be associated and they can thus interact. In other words, we also reconfirmed the system and the validity of this interaction. We then utilized the negative dominance of the protein, meaning that if, as we've seen, this tiny dominion of the protocadherin can interact and can associate itself with the alpha-1 subunit of the GABA receptor, then the idea was that if we remove this fragment, what happens? What we saw was that if we remove or overexpress, and we then allow at cellular level the expression of both protocadherin and of this fragment, which is missing and belongs to the cytoplasmatic dominion at intracellular level, the protein no longer can associate itself with the subunit alpha-1. This is something which is done in vitro, and this system tells us that the two proteins are associated. Now, after that, we also validated these observations at neuron level. The simplest thing we did at first was to check by means of co-localization messages, that is by using an antibody which recognizes protocadherin and an antibody that recognizes the sub-1 alpha-1 unit, and we tried to see whether they co-localize and thus they're associated. Now, if you check the tiny squares, you see that the yellow color is showing the co-localization, that is where the two proteins co-localize. After that, we went to see whether at neuronal levels the two proteins are associated. We did re-carry out the immune precipitation experiments at neuronal level, but also at brain level in a homogenated rat brain. We also reconfirmed and revalidated the assay by using a different assay, which is known as GST pull-down. And we are using the C-term of protocadherin, which was placed in contact with the lysate of rat brain. And we saw that this fragment could capture the alpha-1 subunit of the GABA receptor as well. And then that's when we started wondering why and what is the function of this interaction? What is it instrumental to? So one way to see this is always to modulate the expression of protocadherin at neuronal level. So what we did was using different constructs, and we tried to overexpress at neuronal level the constructs that silence the expression of protocadherin, known as scRNA. And we did use a number of them just to have a twofold control, a double control. We wanted to check whether we truly silenced the proteins. And in parallel, we also used other controls, which we called scrambled controls, sRNA controls. Then we also did a rescue. First, we remove the protocadherin, then we replace it in there. And when doing the different experiments and modulations, we wanted to see what happened to the GABA receptor. And by using an experiment which is known biotechnologies, we wanted to check the levels of GABA in the presence or in the absence of protocadherin at neuronal level. And if we just leave aside this panel and just checking the quantitative results of the study, we saw that in the absence of protocadherin, we have the absence of the expression at surface level of the cell of the alpha-1 subunit of the GABA receptor. Now, obviously, if by abating protocadherin and by just replacing it there, we see that the GABA superficial level goes back to normal levels. The conclusion we extrapolated of the first in vitro data set is that protocadherin interacts with the GABA receptor, and it is also modulating its superficial expression or surface expression. So it is involved in the transportation or traffic of the receptor at surface level. Then we wanted to see what happens in vivo. And what we did in collaboration with Laura Concetta with the ITI was using a faster approach to see in vivo what happens by abating the expression of protocadherin. And we used this in utero electroporation system. So we silenced at ventricular levels in both hemispheres protocadherin. After that, we went to see what happening in vivo. The interesting thing is that in the absence of protocadherin, what we see is a dramatic effect at morphologic level of the neurons. As you can see here, they look as very rachitic neurons. They do not have the branchings. And this is a neuron where protocadherin is missing. So there is a different branching defect and the arborization defect, meaning that neurons are having a defect at migration level. And this is the analysis data. What is interesting, and which leads back to the girl's phenotype, which is epileptic, is that, as we've seen, and we did it in the rats and not in the mouse, the rats are much more susceptible to epileptic seizures. In this case, we gave the PTZ, and the experiment was not conducted by myself, but it was done by the Genova IT. And there is, as we've seen, a latency period after which what comes is the first effect. At this point, we would like to try and carry out some rescues. So that means that we wanted to try some other drugs to see whether we have a pharmacological rescue. And as of now, we are analyzing the phenotype with memory tests to try and see whether the rats also have cognitive defects, which is not only reconducible to epilepsy, but also reconducible to problems, learning problems maybe. And I briefly showed you our conclusions, which are here shown. Protocadherin is expressed at the cortex and hippocampus level in vitro. It interacts and modulates the expression of synaptic markers. And it's also interacting with the GABA receptors while modulating its traffic at superficial level. In vivo, what we see is that we could reconfirm the high expression of the protein, and in vivo lack of the same is creating some neuronal migration impairments. And the subjects are subject also and susceptible to epileptic seizures. But we are also working now in a different directions. We are having a conditional knockout for the protocadherin, and by the end of the year we should have the first crossed model. Right now we don't have any data to produce, but we would very much love to collaborate with you all of you, and work also on PS cells. The laboratory we're working with is also capable of obtaining PS cells and working on them. Right now, our strategy is the one of silencing from controls, i.e. from controlled samples, the protein, and then study the PS cells by differentiating them in neurons, starting off from controls. The laboratory can work the PS cells and utilize them. They are already using it for a different gene as well. And these cells can be differentiated, and they also shown and demonstrated electric activity, which is fundamental to define the neuronal EPS cell. And this is the last slide, the most important one. Here we have those who collaborated in the project. Silvia Bassani is in charge of the entire work of the protocadherin and its characterization. Then we see Laura Concetta. She's in charge of, and Laura Gerosa is also in charge of the cleavage activity of the protocadherin, and Fernando is the student who's involved in this project. Other collaborators were Laura Concetta from the IIT, and as far as the PS cells, and as I said, we don't really have much in our hands. We have had the collaboration of Chiara Bertelli, Carlo Sala from the Neuroscience Institute. Most important thing of all is our sponsors. One is Insieme per la Ricerca with the Acute Syndrome Foundation, the PCDH19 Alliance, and the Cariplo Foundation that funded Silvia Bassani's research, and the BNL that supported us in buying a tool that was very useful for this research. Thank you. Yes, I'd like to ask a question. Have you been using, or have you in mind of using, the unbiased proteomic system to see whether there are any other interactors of the PCDH19? And then, did you observe a presence of PCDH19 within astrocytes? Yes, we did observe it, but we did not investigate any further. We know that it's there anyway. As far as the proteomic, I can tell you in the first place that it's so expensive. Too expensive. But that's an attempt, at least, to have a mapping, because we are working on synapses. We are having, and we're waiting for the animal model. Now, obviously, this is a model which is explaining, or maybe it's not explaining, why is it that in the males we have mutations but no phenotypes are expressed? But there are few cases in males as well. But it's very rare, obviously. That's not the rule. That's very true, very true.